Hello and welcome to Nikolai's genetics lessons, and this video going to be about beginning level of genetics for those people who just start learning genetics. And here's a problem: tall plant height, which is dominant capital allele T, in the P is dominant over the short plant height small t. Question A: if true breeding tall plants are crossed to pure breeding short plants, what genotypes and phenotypes would you expect to see in the F1 generation? And in order to escape confusion, let me go over the terminology first. True breeding here and pure breeding means the same thing. This means that when we take, for example, tall plant, self-pollinated, and in the next following generation, all plants going to be tall. Or if we take a short plant, self-pollinated, in the next generation, all plants going to be the same for this trait. So we call such genotypes true breeding, or such plants, or pure breeding. Pure breeding and true breeding is the same. So once again, true breeding tall plants are crossed with pure breeding short plants. That means that we have to cross genotype capital T with genotype small t. And this is parental generation. In the next generation, we are expecting that all the plants going to be also tall and would have heterozygous genotype. So this parent only can contribute any of these alleles. So this is what we see here. And parent 2 only contribute recessive allele T, which we see here. So phenotypically, these two plants would be tall, but this plant we call true breeding, and this plant wouldn't be true breeding, because if we self-pollinate this plant, in the next generation we would see segregation of traits. Some of the progeny, 75%, would be tall and 25% would be small. So next question: if the F1 plants are selfed, and this is F1 generation, F1 plants selfed, and 1000 F2 plants are obtained, how many of these F2 would you expect to be true breeding when selfed? So basically we have to take plants of F1 generation, and 100% of plants going to be heterozygous, so genotype would be capital T small t. And if we cross these plants, so parent 1 and parent 2 on the side, parent 1 on the top, so what we expect: capital T here, capital T small t here, capital T small t here, and small t here. As you see, 75% of the progeny going to be phenotypically tall, 25% of the progeny going to be phenotypically small, and as you see, this is going to be true breeding, and it would make 25%, and this genotype also would be true breeding and would make another 25%. So we expect that 50% of the progeny of such a cross of F1 generation would be true breeding. If we would cross this genotype with itself, all the plants going to be small. If we cross this genotype with itself, all the plants going to be tall. But if we self-pollinate these plants or self-cross these genotypes, as you see, we are going to get segregation of traits in the next generation. So 50%, as you see, would be true breeding, and 50% wouldn't be true breeding and would be heterozygous. So next question C: a tall plant of unknown history is test crossed, and the resulting progeny are all tall. Draw the cross, including the most probable genotypes of the parents and offspring. What we call test cross. We call test cross when we cross phenotype of unknown genotype with known phenotype and genotype. In other words, for example, tall plants can be capital T genotype and can be capital T small t. So can be homozygous dominant or can be heterozygous, but still these two genotypes would make one phenotype, would be tall plants. And now compare genotype small t and small t. Here, when we take a small plant, we know for sure its genotype. But when we take a tall plant, we are not sure whether this plant is homozygous dominant or heterozygous, whether it is true breeding or whether it is not true breeding. So, how to find if plant is homozygous dominant or heterozygous? We are doing a test cross with known genotype. So we take plant of known phenotype but unknown genotype, and we cross with plant of known phenotype and known genotype. This is what we call test cross. If plant would be homozygous dominant and we cross with a plant that is going to be homozygous recessive, 100% of the F1 generation is going to be capital T small t, going to be heterozygous and going to be tall. So 100% of the F1 generation. But what is going to happen if, for example, tall plant is going to be heterozygous? And we will test cross with short plant of the known genotype. In this case, 50% of the progeny going to be capital T small t, and 50% of the progeny going to be small t genotype. So 50% going to be tall and 50% going to be short. So our test cross would show us that this tall plant of unknown genotype actually heterozygous, because we see segregation of the trait in the next F1 generation. So this tells us that this plant is heterozygous. But in this example, when we do a test cross, we see that 100% of the progeny is tall. That gives us information that genotype of this plant that is tall and unknown genotype actually homozygous dominant. And once again, we can build a Punnett square. In this case, once again, one parent is heterozygous, another parent is homozygous recessive, and if we build a Punnett square, you would see that 50% of the progeny going to be phenotypically tall and going to be heterozygous, and 50% of the progeny going to be short. So 50% going to be tall and 50% is going to be short. Now let's return to our questions. Question D: if the tall plant is known to be heterozygous, what phenotype ratio do you expect in the progeny? And we already gave an answer to this question. We expect that 50% to be tall if tall plant is heterozygous, and 50% going to be short, or ratio going to be 1 to 1. This is answer to the question D. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.